Welcome to Fun Facts Online. There is a saying that has long defined our understanding of autism. If you've met one person with autism, you've met one person with autism. It's a powerful statement that captures the incredible diversity within the autism spectrum, a reality that has challenged researchers and clinicians for decades. Autism spectrum disorder, or ASD, is officially a single diagnosis defined by challenges in social communication and the presence of restricted, repetitive behaviors. But this broad label covers an immense range of abilities and support needs. Now, a landmark study is adding a new layer of biological clarity to this picture, potentially reshaping our understanding of the condition forever. Groundbreaking research from Princeton University and the Simons Foundation, published in the journal Nature Genetics, has identified 4 clinically and biologically distinct subtypes of autism. By analyzing a massive dataset of genetic and behavioral information from over 5,000 children, the study provides a new framework that moves us beyond a one-size-fits-all approach. This wasn't just about looking at isolated traits. The researchers used a person-centered approach, using sophisticated computational models to analyze over 230 different characteristics for each child. This holistic view, covering everything from social behaviors to developmental milestones, allowed them to see patterns that were previously hidden. They found 4 distinct clusters, or subtypes, and crucially, these subtypes aren't just defined by behavior. They are linked to unique genetic signatures. This connection between how autism presents clinically and its underlying biology is a monumental step forward. It helps explain why 2 people with the same diagnosis can be so different. It suggests that autism isn't a single story, but multiple distinct narratives, each with its own genetic script. This research reinforces what many have long understood. The idea of a spectrum isn't a simple line from mild to severe. It's a complex, multi-dimensional condition. This study provides the first robust, data-driven framework that shows there are at least 4 meaningful groups, each distinct in both the clinic and the genome. So let's dive into the 4 biological subtypes identified in this transformative research. The first and largest group is Subtype 1, Social and Behavioral Challenges. This group made up about 37% of the children in the study. Individuals here display the core characteristics we typically associate with autism: significant difficulties with social communication and the presence of repetitive behaviors. But a key distinguishing feature is that these individuals typically reach their major developmental milestones, like walking and talking, right on schedule, similar to their neurotypical peers. Because of this, they are often diagnosed with autism later in childhood, after these early years have passed, without obvious delays. Another defining characteristic of this subtype is a high likelihood of experiencing co-occurring psychiatric conditions. Genetically, this group shows the highest predisposition for attention deficit/hyperactivity disorder, or ADHD, as well as anxiety, depression, and obsessive-compulsive disorder, OCD. The genetic story here is particularly fascinating. For a long time, the prevailing wisdom was that the genetic influences of autism primarily affect the brain before birth. But the Social and Behavioral Challenges group flips this script. The genetic mutations found in these individuals tend to be in genes that become active later in childhood. This aligns perfectly with the clinical picture of later diagnosis and no early developmental delays. It suggests a completely different biological timeline is at play. This group also showed the lowest amount of rare, potentially harmful mutations compared to the other subtypes, adding another layer to their unique profile. Next, we have Subtype 2, Moderate Challenges. This is the second largest group, representing about 34% of the children in the study. Like the first group, these individuals also tend to reach their developmental milestones on time. The primary distinction for this subtype is the severity of the core autism characteristics. They show milder social and repetitive behavior traits when compared to the other groups. But perhaps the most defining feature is what they don't have. Individuals in this subtype usually do not have co-occurring psychiatric conditions like anxiety or depression. This makes them a distinct group from the first subtype, who have similar developmental timelines but very different mental health profiles. The genetic profile of the moderate challenges group offers another piece of the puzzle. These individuals tend to have rare genetic changes, but these variations are found in genes that are considered less essential for fundamental development. This might help explain their milder presentation of core autism traits, as the genetic impact appears to be less disruptive to overall development compared to other subtypes. The third group identified is Subtype 3, Mixed ASD with Developmental Delay. This subtype accounted for approximately 19% of the study's participants and presents a very different developmental trajectory. The defining characteristic here is a delay in reaching early milestones, such as walking and talking. The mixed in the name refers to the variability within this group when it comes to social challenges and repetitive behaviors. Despite the clear developmental delays, a notable feature is that individuals in this subtype are less likely to show signs of anxiety, depression, or disruptive behaviors, which stands in contrast to some of the other groups. The genetic underpinnings of this subtype are also quite distinct. This group was more likely to carry rare inherited genetic variants. These are not spontaneous mutations, but are genetic quirks passed down through generations that may not cause autism on their own, but contribute to the overall risk. This is a significant finding because it points to a different biological mechanism than the spontaneous mutations seen in other groups. The genes affected in this subtype are typically active during early brain development in utero, which provides a strong biological reason for the observed developmental delays. Finally, we come to the fourth and smallest group, Subtype 4, broadly affected. Making up about 10% of the participants, this group faces the most significant and wide-ranging challenges. Individuals in this subtype experience a combination of severe difficulties. This includes prominent developmental delays, significant communication challenges, and pronounced repetitive behaviors. They also have a high rate of co-occurring psychiatric conditions, such as anxiety, ADHD, and other mood disorders. In fact, researchers noted that impulsivity, hyperactivity, and aggressive behaviors were observed only in this subtype, making their clinical presentation particularly complex. The biology of the broadly affected group is the most distinct of all. These individuals had the highest proportion of damaging de novo mutations. De novo mutations are spontaneous genetic changes that appear for the first time in an individual and are not inherited from either parent. These mutations are often more severe in their biological impact, which correlates directly with the more severe and wide-ranging challenges observed in this group. The genetic disruptions here can occur both before and after birth, leading to what researchers termed broad DYS regularity. This group also carries a heavy load of rare mutations in key genes, including some linked to the protein involved in Fragile X syndrome, another neurodevelopmental condition. So what does all of this mean? This research is far more than an academic exercise. It has profound implications for the future of autism, diagnosis, support, and treatment. By connecting clinical presentations to distinct biological pathways, this framework opens the door to the long-sought goal of precision medicine. First, it could lead to more precise and earlier diagnosis. Imagine if a child's genetic profile aligns with the broadly affected subtype. Clinicians and families could anticipate the need for intensive early intervention and support for a wide range of challenges right from the start. Conversely, a profile matching the social and behavioral challenges subtype might prompt proactive screening for ADHD and anxiety as the child grows, allowing for earlier mental health support. Second, this allows for personalized and targeted support. Instead of a one-size-fits-all approach, interventions could be tailored to the biological and clinical profile of the individual. This could mean prioritizing behavioral therapy for one child, while another might benefit more from support focused on motor skills or mental health. Knowing the underlying biology could help families and individuals access the right resources at the right time. And third, it will dramatically advance research and treatment development. Past genetic studies often struggled because they were trying to solve what they thought was a single jigsaw puzzle, when in reality, They were looking at 4 different puzzles all mixed together in the same box. By separating individuals into these more homogeneous groups, scientists can better study the specific biological mechanisms at play and develop therapies that target the root causes of challenges for each subtype. It is important to remember that this study is a foundational step, not a final answer. These 4 subtypes provide a powerful new lens through which to view the autism spectrum, but the human experience is always more complex than any set of categories. These classifications are meant to guide understanding and support, not to place individuals in rigid boxes. The journey to fully unraveling the intricate biology of autism is ongoing. Scientists will continue to refine these subtypes, perhaps discover new ones, and investigate how environmental factors interact with these genetic blueprints. But this groundbreaking research has fundamentally shifted the conversation. It moves us away from a single, monolithic view of autism and toward a more nuanced, accurate, and ultimately more helpful understanding of the many different ways one can be autistic. The blueprint of the spectrum is becoming clearer, and with it comes the promise of a future where every autistic person is understood and supported according to their own unique profile. Thanks for listening to Fun Facts Online.